Hello and welcome to the last section. We'll be talking about section 6 in our video lecture of the digestive system, where we are going to talk about the accessory organs in digestion, including the liver, pancreas, and the gallbladder. So we'll start off just naming what these organs are and what they do. First, we've got the liver here, which is going to help produce bile and export it to the duodenum. Of course, our liver has lots of other functions, but this is what we will focus on. Then we've got our gallbladder here that is going to store and concentrate and release the bile. And lastly, your pancreas, which is going to produce the pancreatic juice, including digestive enzymes and bicarbonate ions, and will then deliver it to the duodenum. So we'll start off with the structure of the liver. It is the largest gland in our body and weighs approximately 3 pounds. We have 4 different lobes here. We have the right lobe and the left lobe. And then if we look at an inferior view here, and I want to orientate you, so this is an anterior side of the liver, and this is a posterior side, and this is again an inferior view here. We are now going to be able to see the quadrate lobe, which is sitting between the gallbladder and the left lobe here. And then we've got our caudate lobe, which is found more posteriorly. So I always remember this by thinking Q&A, quadrate for question, and A for anterior, and then our caudate, that word means tail, so this is posteriorly found where we would find a tail. Then we've got our falciform ligament, which we mentioned when we discussed the peritoneal folds. This ligament separates the right lobe from the smaller left lobe and is going to suspend the liver from the diaphragm, so we would also have a connection up here, and the anterior abdominal wall. Now at the free edge of this falciform ligament, we find a round ligament, also known as ligamentum teres, and this is remnant of the fetal umbilical vein that once was your lifeline. Within the liver too, we have this special area known as porta hepatis, known as the gate to the liver, and you can actually see this well over here where we have lots of different structures entering and exiting the liver. So here we could find the hepatic artery, the hepatic portal vein that will enter the liver, and also the common hepatic duct that is exiting the liver. So the common hepatic duct is leaving the liver because we've created bile through our right and left hepatic ducts and basically channeled it into the common hepatic duct. We then have our cystic duct that drains any bile we have stored in the gallbladder, and where the cystic duct connects with the common hepatic duct, we have the common bile duct, or it could just be called bile duct as well. Now, sometimes if the duodenum does not need that bile there, the liver forms the bile and then it regurgitates kind of through reverse peristalsis and goes back into the gallbladder and can be stored there. I forgot we have this all color coordinated. All right. So this slide here is to kind of show you how you're going to take the nutrient-rich blood and move it within the hepatic portal vein. So in the previous section, we talked about how we are going to have blood vessels supplying the intestines and veins, of course, pulling that fluid, that blood away from the small intestines and the large intestines. So here we have the inferior mesenteric vein. This is going to drain the distal portion of the large intestines, whereas the superior mesenteric vein is draining the small intestines and the proximal portion of the large intestines. Now, we know that as the food moved into our intestines, we were absorbing those nutrients into the capillaries. And so now this blood that we're seeing in these veins is called nutrient-rich blood that is oxygen poor. And so what happens is from the inferior mesenteric vein, we drain that nutrient-rich blood into the splenic vein. and that splenic vein then comes together with the superior mesenteric vein, and all of this nutrient-rich blood goes through the hepatic portal vein and into the liver, specifically making its way into the liver sinusoids. So that brings us to our liver lobules. So we're looking at some of the histology within the liver, and here we could see our liver lobules as being these hexagonal structures and what we call the functional unit of the liver. These are composed of hepatocytes, which you could see those tiny cells here in this illustration, and they help to filter and process this nutrient-rich blood that came in from the hepatic portal vein and made its way through the liver sinusoids that we see pictured here in purple. Now, once we process that nutrient-rich blood, that's going to move into this central vein, which will then connect with an interlobular vein and make its way to the hepatic vein. Now, note that the hepatic veins are not the same thing as the hepatic portal vein. This brought in the nutrient-rich blood. The hepatic veins are going to contain nutrient-poor blood and take that blood and put it into the inferior vena cava to go back to the heart. So notice as we're looking at this liver lobule here, at every corner of where that hexagon is, we are going to have three structures, and this is known as our portal triad. And this portal triad contains a branch of the hepatic artery, which is giving oxygenated blood to our liver. It contains a branch from that hepatic portal vein, which is bringing in the nutrient-rich blood to put into these sinuses. And then we have a bile duct, which is going to take the bile produced by the hepatocytes and move it through the what we call the bile canaliculi, basically meaning these small canals, and moves it into the main bile duct. Now let's talk about the pancreas. This is found mostly retroperitoneal and is deep to the greater curvature of the stomach. In this illustration, the stomach has been removed so you could see the full scale of the pancreas. We know that the head of the pancreas is cradled by the duodenum, and the tail is going to point over to the spleen in the upper left quadrant of the abdomen. Now, the pancreas is really unique in that it has exocrine and endocrine functions. Its exocrine function, meaning it's related to ducts, is going to mean that we are going to produce pancreatic juices, and we do this using the acinar cells, which you could see pictured over here. We've got secretory cells that produce zymogen granules that contain the proenzymes, and then we will move that through the ducts to get its way to the pancreatic duct. This duct, as we said before, is then going to make its way over to be secreted into the duodenum. And we do have some smaller duct cells that will produce water and bicarbonate in order to neutralize the acidic chyme that could enter the duodenum. The endocrine function of our pancreas is going to be its ability to secrete insulin and glucagon by these pancreatic islet cells. So first, before we get into the endocrine processes, let's talk about the pancreatic juice. The juice is going to be a watery alkaline solution with a pH of 8 to help neutralize that acidic chyme coming in from the stomach. So will contain within it electrolytes, and the primary electrolyte will be our bicarbonate ion. Other digestive enzymes will contain proteases that will break down proteins. They're secreted in the inactive form in order to prevent self-digestion. Then we've got pancreatic amylase that will break down our carbohydrates, our pancreatic lipase that will break down lipids, and our nucleases that will break down nucleic acids. Now let's talk about the gallbladder. The gallbladder is going to be a thin-walled muscular sac on that ventral or anterior surface of the liver, and its job, once again, is to store and concentrate the bile by absorbing water and ions. We look inside the gallbladder and can see this honeycomb fold that will allow it to expand as it fills. We also have muscles within the wall here so that it can contract and release that bile through the cystic duct and flow into the common bile duct that will then go through the hepatopancreatic ampulla and into the duodenum. Now, what is bile made up of? It is this yellow-green alkaline solution that is going to contain bile salts. And our bile salts is a cholesterol derivative that is going to function in emulsifying fats and absorbing it. We also have bilirubin in it, which is going to give the pigment, and this is formed from the breakdown of red blood cells and taking that heme. The bacteria break down in our intestines, will then turn it into stercobilin that gives that brown color to our feces. And just a note that we are not going to talk about section 7, which is on chemical digestion and absorption.